Hello, my name is Samuel Chen, and I am an undergrad studying computer science at Michigan State University. I am doing research under Dr. Janani Ravi, and today I will be presenting our work on MolEvolvR, which is a web app used to characterize proteins using molecular evolution and phylogeny. Molecular evolution and phylogeny can be useful tools in investigating pathogenic protein families. By studying how these proteins evolve across bacterial lineages, we can identify lineage-specific signatures and variants, and consequently, their functions. We have developed a streamlined computational approach for the molecular evolution and phylogeny of target proteins, and we have implemented this approach in our MolEvolvR web app. The approach we are using starts with our query protein, and we characterize these proteins by domain architectures and genome context. Characterizing by domain architectures means looking at the proteins by their constituent domains, and characterizing by genomic contexts means looking at all the proteins that make up the genomic neighborhood. We then perform homology searches to obtain thousands of protein homologs, and we perform the same kind of molecular characterization on these homologs. Finally, we are able to run some custom R scripts to summarize this data in terms of phyletic spread, proximity networks, and structure-based sequence alignments. This computational approach is implemented in our MolEvolvR web app. Here is a screenshot of the app, and users can upload their protein data at various stages in our approach. For example, the app accepts accession numbers, the results from homology searches such as BLAST, as well as results from IntegroScan. Once we run this data through the process, we can analyze and visualize the protein data by the domain architectures. Some of the ways in which we can visualize the results of our analysis include this upset plot, which shows the domains that co-occur and in which combinations, as well as the frequencies of these combinations. This heat map shows the phyletic spread of our domain architectures, and using this, we can easily identify how domain architectures have evolved across lineages. Finally, we can view the data in a domain proximity network, and in this domain proximity network, the nodes represent domains, and edges between the nodes signify that the domains occur adjacent to each other. Using this network, we can easily identify which domains co-occur and interact with each other, which allows us to better identify function. Our approach gives us information about the phylogeny of the target proteins, and one of the visualizations we can generate include phylogenetic trees. Here we have generated a phylogenetic tree for one of our target proteins with the lineages colored by phylum. To the right, we have generated a similar tree, only this time with their multiple sequence alignments. These plots provide insight into how the protein has evolved and been conserved across lineages. These sunburst plots display each of our target proteins and their fractional prevalence in each lineage. To summarize, this approach enables us to study molecular evolution and phylogeny in a much more streamlined and automated fashion. Ultimately, this can help biologists identify lineage-specific pathogens, which could be useful for finding diagnostic or treatment targets. Additionally, examining proteins through the lens of domain architectures provides a more rigorous analysis than if we only used the protein sequence. For the future, we are planning on releasing an R package such that computational biologists may use them with their own data, and additionally, we plan on implementing genomic context characterization of proteins in MolEvolvR, which will provide an even more rigorous analysis when applied to bacterial proteins. I would like to acknowledge Janani Ravi, Lauren Sosinski, the entire JRavi lab, and GLBIO for providing me the opportunity to talk. Thank you.